Hello, this lecture is entitled Micro 101, and I'd like to spend a little time talking about some of the major classes of bacteria and bacterial infections. For the purposes of this discussion, viral infections and fungal infections will not be included. Basically, you can think of bacterial infections in three different classifications: gram-positive coccal infections, gram-negative infections, and anaerobic infections. And most of the bacterial infections that we see come into one of those classifications. Some are mixed and include bacteria from from all of those particular categories. But it is helpful to think about bacterial infections in this way, especially when it comes to picking the sorts of antibiotics for treatment. Let's talk a little bit about gram-positive cocci. The main ones we're talking about are streptococci and staphylococci. Streptococci can be further categorized as alpha hemolytic, beta hemolytic, and gamma hemolytic, depending on their ability to hemolyze sheep red blood cell agar. The alpha hemolytic streptococci are also known as strep viridans. Comes from the Greek verdant, green, because it produces a green partial hemolytic zone on the sheep red blood cell agar. Most of these bacteria that are alpha hemolytic are in our salivas and can occasionally get into the bloodstream, and the most notorious infection from strep viridans when it gets into the bloodstream from dental work is endocarditis. Pneumococcus, which comes from a human host and can reside in our nasopharynx, is also alpha hemolytic. The other subcategory of streptococci are beta hemolytic streptococci, and these can be found in the nasopharynx or the upper respiratory tract of even healthy individuals, as well as laying on the skin of otherwise healthy individuals. And these bacteria, the beta hemolytic streptococci, are frequently involved in skin and soft tissue infections and are the main culprit for non-pyogenic cellulitis. Group A streptococcus is otherwise known as strep pyogenes. Group B streptococcus is otherwise known as strep agalactiae, and you can go all the way down the alphabet to strep G. I don't know if there's any bacteria further than that letter. But basically, these produce a clear zone around the sheep red blood cell agar and are again referred to as beta hemolytic. Then the third subcategory of streptococci are gamma hemolytic strep, and probably the most important one of this is Enterococcus. Enterococcus, as the name implies, is found in high concentration in the small bowel. Think of it as the small bowel cocci. And if the bowel is disrupted, of course, it can cause intra-abdominal infections. And since it is excreted in the stool, it is a relatively frequent cause of urinary tract infection. And again, it produces no zone of hemolysis whatsoever on the sheep red blood cell agar. Those are the three main types of streptococci. Moving along the continuum of gram-positive coccal infections, we have staphylococci. There are two types of staphylococci that I want you to know about. One is coagulase-negative staphylococci, which staph epidermidis, and most other staphylococci that reside on our skin are. The gram coagulase positive staphylococci are the more virulent staphylococci and are also referred to as Staph aureus. So all coagulase negative staphylococci are Staph epidermidis, and variants of other coagulase negative staph include Staph hominis, Staph saprophyticus, and there's numerous other coagulase negative staphylococci. The gram-positive staphylococci, Staph aureus, there are really only just two types of Staph aureus. One is methicillin sensitive and one is methicillin resistant. When you look at the antibiotic gram of staphylococci and what antibiotics it's sensitive to, you will not see methicillin on that list. Instead, it has been supplanted by oxacillin. The reason for this is that the methicillin disc in the laboratory was very perishable, and the oxacillin disc was more stable, and hence it made it to the practical laboratory. However, the nomenclature has not, and if the organism, if the Staph aureus is sensitive to oxacillin, it can be considered methicillin sensitive Staph aureus. If it's resistant to oxacillin, it will be referred to as methicillin resistant Staph aureus. And incidentally, the same applies to coagulase negative staph, which can be oxacillin sensitive or oxacillin resistant, rendering the names of these organisms methicillin sensitive Staph epi and methicillin resistant Staph epi. When it comes to looking at the blood cultures, in the blood cultures that we use here at Mercy Fitz, there is a relatively new technology that checks for the genetic material in the blood and further helps us distinguish between Staph epi and Staph aureus. The MEC gene notation that is on the biophore simply refers to whether or not the staph is methicillin sensitive or methicillin resistant. The MEC gene confers methicillin resistance, but this has nothing to do with whether the organism is coagulase positive or coagulase negative. To have an methicillin resistant Staph aureus, as notated on the biophore, this requires the MEC and the MEJ genes. If you have both of them, you have the makings of MRSA in the bloodstream. But if it's just the MEC, that can apply to Staph epi or Staph aureus. Hopefully I've made that clear. The next, and I should also say that Staph aureus infections are typically skin and soft tissue infections. When it does cause cellulitis, it's typically pyogenic, especially if it's MRSA as opposed to streptococcal cellulitis, which is non-pyogenic. So staph can be found in the environment, it can be found on our skin, it can be found on our nasopharynx, but in general, it will cause soft tissue infections. On rare occasions, it can cause respiratory tract infections, and of course it can get into the bloodstream and cause bacteremia and wreak all kinds of havoc in the body, which we'll discuss later. The second major category of bacteria are gram-negative rods. These exist in nature, especially Pseudomonas aeruginosa, which loves water, and as we see quite often is involved in foot infections and nail puncture infections because it happens to reside in high levels in our shoes and socks and so forth. But most of the gram-negatives that we deal with are actually in our guts, especially the distal small intestine and also to some degree the colon. Therefore, they become important causes of intra-abdominal infections, ruptured viscuses, cholecystitis, diverticulitis, and also urinary tract infections, especially urinary tract infections, because we tend to get infected with our for urinary tract infections with our fecal flora. So again, gram-negative infections are usually seen in intra-abdominal and urinary tract infections, generally speaking. I should also note, however, though, that after a person is in the hospital or in the nursing home for some time, in addition to the usual strep and anaerobes that can be found in the mouth, gram-negatives and MRSA for that matter will populate the saliva and can be involved in hospital-acquired aspiration pneumonia. But typically, outside of the hospital, gram-negatives are seen as frequent causes of urinary tract infections and intra-abdominal infections. The third group of organisms are anaerobes, and they can be thought of in two separate categories. One that are above the diaphragm, because anaerobes can be found in high quantities in the saliva and the mouth. And therefore, if we aspirate our saliva, are frequent pathogens in aspiration pneumonia. Most of these anaerobic bacteria are anaerobic streptococci, Peptostreptococcus, Peptococcus. There are gram-positive anaerobes in the mouth, including other than streptococci, including Eubacterium and Bifidobacterium. Much less common organism that we see are actinomyces, which are anaerobic, which can cause mouth infections and chest infections, presumably from originally derived from the saliva. Anaerobes below the diaphragm are primarily B. frag, Bacteroides fragilis, which occurs in high concentration in the colon and is involved in, along with other organisms, in intra-abdominal infections, especially the gram-negative infections, and interestingly is not involved in urinary tract infections because the urinary tract cannot support the growth of strict anaerobes. And that's because urine itself has a high content of dissolved oxygen and cannot sustain growth of anaerobes. There are a number of infections that involve mixed flora, including gram-positive, gram-negatives, anaerobes. These infections include aspiration pneumonia because the saliva contains both anaerobes and aerobes, and as such, antibiotics need to be given that will cover multiple classes of these particular bacteria. Diabetic foot infections will frequently be polymicrobial and involve anaerobes, streptococci, gram-negatives, and broad-spectrum antibiotics that cover all these classes need to be used as well. Intra-abdominal infections can involve all three classes of bacteria, including gram-positive cocci, Enterococcus, gram-negative rods, E. coli, Klebsiella, Serratia, Proteus, Citrobacter. All of these are gram-negative rods, and strict anaerobes, especially B. frag, for diabetic and other anaerobes such as non-fragilus Bacteroides, and these are involved in diabetic foot infections as well and intra-abdominal infections. So when thinking about infections and diagnoses and covering with antibiotics, you should think in these broad terms of could this be a gram-positive infection, could this be a gram-negative infection, and could this be an anaerobic infection or even a mixed infection, and therefore what antibiotics should be used. I'll go over this very slightly right now because we'll talk more about antibiotics in the future. If we're dealing with a soft